You know, in medicine, we're almost always chasing perfection, right? Perfectly designed molecules, perfectly targeted therapies. But what if the real secret to a revolutionary new treatment isn't perfection at all, but actually a little bit of chaos? We're about to dive into a fascinating discovery that might just flip how we think about delivering medicine to our cells.
And that brings us to this really critical question that scientists are starting to ask. When it comes to getting medicine inside our cells, could our obsession with making the perfect package actually be holding us back?
To really get why that question is such a big deal, we've got to look at this incredible technology that's at the heart of so much of modern medicine: lipid nanoparticles.
So what exactly are they? Well, think of them like microscopic delivery trucks. They're these teeny tiny bubbles of fat, or lipids, that are designed to carry really precious medical cargo, like the mRNA from the COVID vaccines, safely past all our body's defenses and right into our cells.
And their importance? I mean, it is huge. A lot of us first heard about them because of the COVID-19 vaccines. But that's really just scratching the surface. Scientists are using these exact same tiny vehicles for things like groundbreaking cancer treatments and even therapies for rare genetic diseases. Their potential is just massive.
But there's a problem, a really big one that's been kind of a roadblock for their potential. For years, scientists have just been stumped by what they call the 1% delivery problem.
Get this, 1 to 5%. That's it. This is the shockingly low percentage of medicine that these nanoparticles carry that actually makes it to its destination inside our cells.
Yeah, you heard that right. The vast majority of the cargo, we're talking over 95%, it never gets released. It just stays trapped inside its little delivery truck, totally useless. And you can imagine this has been a huge bottleneck for developing more effective treatments.
And as Artu Brewer, the lead researcher on a new study, points out, this inefficiency isn't just a lab problem, it has major real-world consequences. I mean, think about fighting something like fast-dividing cancer cells. If you deliver too little medicine, the disease can simply outrun the treatment. Solving this delivery problem is, quite literally, a matter of life and death.
But now, something's changing. A really surprising discovery is shifting our whole understanding of this problem. And it all started because a team decided to look at these nanoparticles in a completely new way. So, instead of just looking at the average properties of a whole batch of these things, Brewer's team came up with a way to analyze 1 million individual nanoparticles, one by one. And what they found was that these particles, well, they weren't all the same, not even close. They found 2 totally distinct types of nanoparticles in every single batch. First, you had the organized ones, where the cargo was packed all neat and tidy. You know, the kind everyone had been trying to make for years. And then there were these amorphous, or messy particles, where the cargo was all disorganized. And here's the huge surprise. The messy ones were way more effective at delivering their medicine.
I know, right? It's completely counterintuitive. For years, the whole goal was perfect tight packing. So why on earth would a disorganized, messy particle work better? Well, the answer is actually kind of beautiful.
See, in those perfectly organized particles, the positive and negative charges of the lipid and the medicine are locked together super tightly. They're actually too stable to break apart. But in a messy particle, those charges are just a little bit separated. And that's the key. Once it gets inside a cell, that little bit of separation allows the positive charges to suddenly push away from each other, literally blowing the particle apart and releasing all that medicine. So, okay, let's walk through it. First, that messy particle gets inside a cell. The environment inside the cell is different, and that triggers a change. All of a sudden, those positive charges start to strongly repel each other, and boom, just like that, the particle falls apart, dropping off its cargo exactly where it needs to be. It's basically designed to fail, but in the best possible way.
This discovery isn't just some minor tweak, you know? It really represents a fundamental shift in thinking. It points us toward a completely new direction for how we develop drugs.
The goal has completely flipped on its head. Instead of trying to create these perfectly stable, organized nanoparticles, the new objective is to intentionally design disorganized ones, particles that are stable enough to make the trip through the body, but unstable enough to break apart right when they arrive. And that quote really drives home just how radical this idea is. For years, the entire field has been running full steam in one direction, chasing this idea of perfection. And now, this research suggests the most promising path forward is in the exact opposite direction. Which leaves us with a pretty fascinating thought to end on. We started by asking if perfect was the enemy of good. Well, it turns out it just might be.
In a field that's built on precision and control, maybe, just maybe, the next great leap forward will come from embracing a little bit of beautiful, functional chaos.